Two central nervous system (CNS) centers known to control micturition are 1. Pontine micturition center, PMC, location, located in the brainstem, specifically in the pons. Function, it coordinates the detrusor muscle contraction and external urethral sphincter relaxation required for voiding. 2. Periaqueductal gray, location, found in the midbrain around the cerebral aqueduct. Function, acts as a relay and integrative center that processes sensory information related to bladder fullness and sends signals to the PMC to initiate micturition. These centers work together with cortical and spinal pathways to regulate voluntary and involuntary control of urination. Micturition, or the act of urination, is controlled by a complex interaction between the central nervous system, CNS, and the peripheral nervous system. Here's a detailed explanation of the two CNS centers that play key roles. 1. Pontine micturition center, location, situated in the pons, a part of the brainstem. Role in micturition. The PMC is the primary center responsible for initiating and coordinating the act of micturition. When the bladder is full and the decision to urinate is made, signals from higher brain centers like the prefrontal cortex reach the PMC. The PMC then orchestrates the following actions. Activates parasympathetic nerves to contract the detrusor muscle, the bladder wall. Inhibits somatic motor neurons controlling the external urethral sphincter, causing it to relax. These actions work together to empty the bladder. 2. Periaqueductal gray, PEG. Location, found in the midbrain, surrounding the cerebral aqueduct. Role in micturition. The PAG functions as a relay and integrative hub for sensory information from the bladder. It receives signals about bladder fullness from stretch receptors in the bladder wall via spinal pathways. Based on this input, the PAG communicates with higher brain centers to assess whether it is an appropriate time and place to urinate. If so, it sends signals to the PMC to initiate micturition. Bladder filling phase. Sensory information about bladder fullness travels to the spinal cord and is relayed to the PAG. The PAG keeps the PMC inhibited while the bladder fills to prevent involuntary urination. Micturition phase. Once the bladder is full and socially appropriate conditions are met, the PAG signals the PMC to activate the voiding reflex. The PMC coordinates the detrusor muscle contraction and sphincter relaxation to allow urination. This coordination ensures a balance between involuntary